Good morning, everyone, and welcome to the 2014 edition of the Royal Tyrrell Museum Speaker Series. Today, the Royal Tyrrell Museum and its cooperating society are proud to present Dr. Kevin Campbell. Kevin is a professor in the Department of Biological Sciences at the University of Manitoba in Winnipeg. Kevin was born here in Taber in Alberta, but grew up in Morden, Manitoba. He obtained both his bachelor's degree and PhD in biology at the University of Manitoba. For his dissertation, he studied the nutritional ecology and bioenergetics of muskrats. After completing his PhD, Kevin completed a postdoctoral fellowship at the University of British Columbia in Vancouver and a second postdoc at the University of Aarhus in Denmark, studying hemoglobin and myoglobin in more details. It is during his years spent in Denmark that he began a study on woolly mammoth hemoglobin, a project that he continued as a professor at the University of Manitoba and took over 9 years to complete. Kevin's research interests focus on the physiology of extinct and living mammals. In addition to the woolly mammoth, he has started to study the DNA of mastodons, stellar sea cows, and woolly rhinos, looking at over 100 genes to learn about the development, neurobiology, tooth development, and thermal biology of these extinct animals. When not working on extinct species, Kevin focuses on the respiratory physiology of moles and shrews, two groups of mammals with special blood adaptations for oxygen delivery. Today, Kevin will present an overview of his work on woolly mammoth DNA in a talk entitled, yeah, I got that right, Back from the Dead, Resurrecting Physiological Traits from Long Extinct Species. So without further delay, I present you Dr. Kevin Campbell.
Many thanks for the introduction, Francois. It is kind of a homecoming. I was born in Alberta, and I actually moved away when I was very small, about 6 months old. But it is nice to be back in familiar territory, I guess. And it's also, I have to say, I've given quite a number of talks, but I've never given one in such a lovely venue, in such a topical venue. To see the fossils right here is beautiful. And I live in Winnipeg, and this is what Winnipeg looks like still. So take heart. Except for we've got more snow in Winnipeg. And when I did the first interview on this work a few years ago, and it was in May, and it was snowing, and the reporter, was glum about it. I said, "It's perfect. It's so topical. So take heart. Today's snowfall is again just right into my hands. It's just ideal for us. It's going to talk about this because I'm going to be talking about, cold in this, and I'm going to be talking about the genesis of an idea that came about actually in early 2001, and it took many years for technology to catch up. And now the technology has gone just so far beyond, and I'm going to finish off the talk talking a little bit about that technology and where we're going to go from here. Right now, I can tell you, I've already generated far more data than I can possibly go through in my entire career. I've got a good 20 years left, I hope, and there's no way I can touch it. Right now, they're producing thousands and thousands of times that information in just days. It's really quite remarkable. So I studied muskrats as my PhD, moles in a postdoc in mammoths. So I'm stuck on M's. So I'm going to talk a little bit about the past. Because as it turns out, every organism on Earth here today is really the culmination of the past. And one thing we know, especially talking to paleontologists, I think they're more familiar with than anybody, that the Earth is not a static place. We keep talking about global warming, about climate change. Well, the entire Earth's history has been climate change. Just take a look here. 18,000 years ago, this was snow coverage in North America. So this is a circumpolar view, looking down at the North Pole. Today we've got Greenland here, but 18,000 years ago, just look at that ice cover here. And when we look at physiological adaptations of animals across any kind of spectrum, we like to think of adaptations, right? Birds are adapted to flying. Feathers are adapted for flying. Well, that's actually not why they arose. Feathers actually arose, well, to keep them warm. And they took on this new change. So the climate is constantly changing, the Earth is constantly changing, continents are moving, and animals have traits that we like to think may be adaptive or beneficial to them, but a lot of those traits actually evolved eons ago in completely different environments and have just simply been maintained. So we don't really know sometimes whether something's adaptive or if it's just something that just still happens to be there and may still be useful. Oops. And one way to actually look at this from a different perspective, as I mentioned, we are the result of eons and eons of evolution. And our genomes actually hold all kinds of information regarding our distant past. Not even just our recent past, but our distant past. And there's all kinds of, like yesterday in Nature, there was just a beautiful article, I know it's open to anybody to read, talking about even just humans and interbreeding with Neanderthals. And so now they can look at anybody's genome, and they can pick out the little fragments in your genome that used to be in a Neanderthal. Right? So we still have that information. Genes and proteins carry historical information. Well, in a museum like this, we also have all kinds of paleontological information, geological information. And these all together give us 3 independent records of the history of life on Earth. Whoops, sorry about that. So it's one thing to look at it just from a geneticist viewpoint. It's another thing to look at it from just a paleontologist viewpoint or a geologist. But actually now what we can do, and the way science is really moving forward, is nobody's working in isolation now. With the internet, with Skype, all these factors, we can be in immediate contact with collaborators all over the globe, transferring all kinds of information instantaneously. And so there's all kinds of interdisciplinary work that I think is really going to be the future. So where we can look at, let's say, for my interest, a protein within the context of its planetary biology. Because a lot of people actually, they look and say, oh, I see this animal here, I'm going to study it. Oh, it must be adaptive for living in this environment. Let's say this muskrat in this marsh. This trait has to be adaptive because it's living here. Well, if you go back in time, you know, I think there's all kinds of ways to look at these things differently. And I'm going to try to show you a few of the ways that we can do that. And I got really interested in studying hemoglobin. Hemoglobin is this fantastic molecule that resides in your blood. It actually gives your blood that red color. And hemoglobin is interesting, and it's under a lot of selection for all kinds of animals because it is the single molecule that unites your body with the atmosphere. Hemoglobin was one of the first proteins together with myoglobin to have its crystal structure deduced, and I'm going to talk a little bit about that in a few minutes. And it was awarded the Nobel Prize by a guy named Max Perutz, and he called hemoglobin the molecular lung because it picks up oxygen at the lungs, and then it breathes, it changes shape, and it physically opens up the molecule a little bit, and it releases oxygen at the tissues. So it's kind of a living molecule that changes shape as it performs its functions. And if you look at vertebrates, going all the way back from the earliest fishes up to birds and mammals and reptiles, hemoglobins are a tetramer. That means they're a 4 subunit molecule, and it's encoded by 2 alpha globin chains, you see here in the yellow and green, and 2 beta type globin chains, and they're fairly small by protein standards. So the alpha chain has 141 little amino acid building blocks, whereas the beta chain has 146. And I like to call hemoglobin myself the molecular Robin Hood. Because what it does is it steals from the rich at the lungs, it changes conformation, and it picks up oxygen where there's lots of oxygen available, and automatically, once it gets down to the tissues, it senses that there's very little oxygen around. And it kind of feels bad for them. And what does it do? It changes its structure, and it offloads that oxygen to the tissues. So this is a very important molecule under a lot of selection for animals that live underground, animals that dive, animals at high altitudes, and as it turns out, animals in the cold. because it links oxygen in the environment to the oxygen going to your cells or to the animal's cells. And we like to think of hemoglobin as just being oxygen-centric. As it turns out, it is a remarkably complex molecule that does many other features. I have a friend, Joe Bonaventura, down at Duke, who's been studying hemoglobin for about 40 years. And he studies mostly these sorts of things. And in his talks, he talks about those things, and he goes, oh, and by the way, it also binds oxygen. So it plays key roles in the delivery of carbon dioxide in the body, but it doesn't really bind too much of the carbon dioxide directly. It carries protons, which are the acid, and it actually is your blood's primary buffer system for pH management. And it turns out there's recent work now that shows that it plays a key role in nitric oxide uptake and delivery. And nitric oxide is a miracle molecule that does all kinds of things in your body. It's just remarkable things. And one of its key things is it actually opens blood vessels. And it kind of tells your blood that we need more oxygen here. Well, as it turns out, hemoglobin carries that molecule, and it actually seems to, now we understand, opens and closes blood vessels itself. So it directs blood to where it needs to go. And I'm going to talk about two other molecules. I'm going to talk about these two guys, 2,3-DPG, which is only found inside red blood cells, and it binds to hemoglobin. It plays a key role in its ability to bind oxygen and release it appropriately, as does simple chloride, right? We know this from table salt, sodium chloride. And there's a lot of that in your blood. So those are going to be some key players today. So, as I mentioned, temperature plays a role. We all know right now with winter, we don't see snakes. We don't see any lizards out. It's simply too cold. They're ectotherms. And even in the summer, in the morning, these ectotherms are kind of sluggish. Their metabolic rate's low. So temperature plays a huge effect on all kinds of enzymatic or all kinds of metabolic reactions. So at low temperatures, things go really slow. So there's a certain energy threshold that needs to be kind of met for chemical reactions to occur. So at cold temperatures, as it turns out, things don't work as well. And that's the same for hemoglobin. And this is because to break that little weak bond between oxygen and the heme groups, you need energy. You need heat. And at cold temperatures, there's less energy. So as temperature drops, hemoglobin becomes more and more selfish. It holds on. It binds oxygen tighter. And this has very negative ramifications for, as it turns out, humans in the cold. And not just humans, but all kinds of animals that aren't clever enough to, knit nice little mitts, toques, put on boots. So here, this is just some data from 1955, and a lot of this early thermobiology work was being deduced on reindeer and on muskox and even on these big sled dogs. So one thing we see here, if we look at animals, unlike ourselves, they are what we call regional heterotherms. And they allow their extremities to cool dramatically. And this is magnificent. This is a huge, huge benefit to these animals. So how do they do this? Is they have what we call countercurrent heat exchangers. It's a very simple, basic plumbing system whereby arteries carrying hot blood going down to the extremities runs anti-parallel, really close to veins coming back. So as blood moves down, it transfers heat directly to the cold blood coming back. So instead of losing an enormous amount of heat to the environment, the heat is actually short-circuited. And they have constant blood flow to the extremities. That's very different than what humans do. We're not adapted to cold at all. Our ancestry is from, sub-Saharan Africa. And what we do to minimize some heat loss is we simply just turn off blood flow. And our extremities get really cold. And when things get dangerous, if you're stuck on Everest or you're stuck, your body kind of goes, I'm going to kind of keep things in the core. And we just cut off blood flow, and that's where people get frostbite. Fingers will freeze. And there's another problem with this in humans because our blood doesn't work well in the cold. So even if we do put some blood there, no oxygen's being coming off of the hemoglobin. Because in us, we get what I would call a supply-demand mismatch. So this is some curves. I'm going to show you a few curves later on, but I'll just briefly explain here. This is basically the amount of oxygen. So as you have more and more oxygen present on the x-axis here, if you look at the y-axis, this is the percent saturation of the hemoglobin. So as you have more and more oxygen, it's highly saturated. As you have less oxygen, very little saturation. And here's two graphs for human hemoglobin at 30 degrees Celsius and at 10 degrees Celsius. So one thing you see, as temperature goes down, the affinity of the molecule goes up substantially. And right here around 40 millimeters of mercury, that's about the amount of oxygen that is found at your tissues. Right? That's in the blood. So here we can see in you and me, you guys sitting there right now, as blood is moving through your legs and through your arms, it's actually only offloading about 25% of its payload when you're at rest. When you exercise, you can offload more. But right now, you're only offloading about 25% of the oxygen as it's moving through your body. Well, if I were to lower your temperature of your fingers down to 10 degrees Celsius, hemoglobin is only able to offload maybe 3%. So we're having an ability of hemoglobin to offload. Its ability to offload, dropping from 30 to 10, it goes down between 97 and 100%. But as it turns out, cold tissues need less oxygen. Right? A cold lizard is consuming less oxygen than a warm lizard. But demand goes down about 75%. So we see that these two variables are not matched to one another. Supply goes down much faster than does the demand. So what's the solution for this? Well, if I'm an animal like a sled dog or a reindeer or a muskox that's standing on ice and cold, and my feet are cold for 6, 7, 8 months straight, we can't have no oxygen for that period of time. The tissues will die. Right? Again, that's what happens to us. Right? So what have they done? They've solved this. They've modified their hemoglobins through evolution to have what we call a low oxygenation enthalpy. And here's some curves for cow hemoglobins. And again, we find these in cows, but this is something that actually evolved, we're pretty sure, during the Oligocene, during the initial major Earth cooling event in the Oligocene. And it's found in all ruminants, including muskox and reindeer. But it's a hangover. It's got evolutionary hangover that's found in all ruminants, even those that are found non-Arctic environments. So we see here, temperature has a much, much less effect on the oxygen affinity of the blood. And what this allows here is for demand to match supply. Well, how do we really look into this and see, is this adaptive? Right? I find it in cows, and it seems a little strange because you don't see cows up in the Arctic. Well, a good way to test this kind of hypothesis is to look for animals whose ancestors were in the tropics, and then they moved to the Arctic. As it turns out, the penungulates, which include the elephants, the hyraxes, and the sea cows, are ideal model systems in this because they evolved, all of them, in Africa about 80 million years ago. And Africa, 80 million years ago, like today, was a very, very warm place. The Ice Ages, Africa was still a tropical, warm place. However, we have three groups here, I show in blue, that very recently, very abruptly moved into high latitudes. And let's look at this. I'm going to show you that this was a very strong selection pressure, and we know this. Let's take a look here. An African elephant is a cousin of woolly mammoths and Asian elephants. Okay? So take home message number one. Woolly mammoths are poorly named. They should be called woolly elephants because they're elephants. During the ice ages, we had massive drops in global temperatures. So what we see here is average Earth temperature over about the last 200 million years. So during the age of the dinosaurs, it was much, much warmer than today. And then around the Oligocene, we had this huge drop in temperature. And then during the ice ages, massive reductions in temperature, strong selection pressures. And we know this was the case because mammoths are woolly. The number one physiological change or challenge for an elephant is dissipating heat. They're huge. They've got these great big ears. They've got these thermal windows. Their number one challenge is getting rid of all this metabolic body heat. Well, for mammoths, we knew the tables immediately turned within only the last 1 to 2 million years as they moved to the Arctic because their ears got small, their tails got shorter, and they developed this thick, woolly coat. So they wanted to conserve heat. And how do you conserve heat? The best way to conserve heat is to have a countercurrent heat exchanger. Conserves heat, and it reduces your energy bill. You need to eat less food if you're producing less heat when there's less food available, when it's dark, when it's wintertime. And again, I want to highlight this challenge here and where we find most of these mammoths. So just looking at 18,000 years ago, most of the mammoths were found in Alaska and in Siberia. It was a desert. So while we might have 800 meters or so of ice and snow across most of North America and Europe, up here in high Asia, Siberia, was a bit of a desert, the Mammoth Steppes. Very productive land, but very little snow. And as it turns out, a fair amount of permafrost now that has preserved animals such as woolly mammoths that went extinct. It's about 3,700 years ago on this tiny little island, Wrangel Island. The mastodons from North America, maybe 12,000 years ago. And finally, one that we're working on is super exciting is something called stellar sea cow. And these guys lived way up in the Arctic here. They were discovered in 1741 and hunted to extinction by 1768. So we've got 3 perfect model systems to look for evolution of cold adaptive traits independently in different lineages, from tropics to the Arctic. Okay? Are you agreeing with me? These are ideal model systems to look at physiological blood oxygen delivery. How do these things work? But hopefully you've seen the hole here, the stumbling block. And I show this picture because this picture was my inspiration. I was in Denmark and I was watching a TV show about this mammoth. This is the Yarkov mammoth. And they said they found this huge full-body mammoth and they were going to do it and they were going to clone it. This is December 2000, January 2001. This is what they found. They were going to clone this animal 14 years ago. I don't see any walking around yet. But that was what it was. And the next day I came to work and I was sitting in the coffee room and I was looking at this magazine. Actually, I had a dinosaur on the front. And I was going, oh, that's so cool. And I was thinking, what else is cool? And I went, well, mammoths are cool. And the idea just hit me at that moment. But I work with blood. I don't see how I'm going to put a syringe in there and get my little blood sample. And even if I did, it would be hopelessly degraded. That's what we're saying. I mean, this guy here, 38,000 years or so, it's just degraded. It's all there is to it. It's no good. But we're physiologists, and like I say, why would we let something being dead stop us? Let alone just being dead for 30,000 years. Because these animals have so much going for them. Look at them. They're huge. They've got unbelievable fossil records. We know, without any ambiguity at all, mammoths evolved in Africa about 7 million years ago. And for 5 million years, they did not leave Africa. There's all kinds of mammoth fossils because their teeth. We can see these things. We can see their bones. Excellent fossil history. We know precisely who they're related to. Very recent divergences. Okay? 6 to 7 million years ago for a large animal like that is a blink of evolutionary time. And these large animals evolve much slower than small animals. So we would think that there's very few genetic changes between an elephant and a mammoth. And at the year 2000 or early 2000s, mammoths were the only species which ancient DNA had managed to get a tiny little snippet, about 90 base pairs of nuclear DNA. Your genome has 3.2 billion base pairs of nuclear DNA. By the year 2000, 2001, one person had gotten 90 base pairs from a mammoth. So I phoned him and I talked to him about this, and I kind of suggested what my idea was, and he thought maybe we can get this done. Just these two little tiny genes, right? He thought, yeah. So what you need is some mammoths to do this, of course. And where do we find these mammoths? Extreme north. Take a look at this. I mean, if there's any place in Siberia that you're going to think is going to be cold, well, these would be the places. 73 degrees north, 72 degrees north, 70 degrees north. And this one here actually turns out is the one we primarily use. This is a femur bone dated to about 43,000 years ago, magnificently preserved DNA in it compared to any of the other samples. So this was our primary sample. So we were thinking, yeah, these guys were really cold. And we did the work, strangely, in Australia. Two reasons for that, because they had opened this brand new Australian Centre for Ancient DNA. This was Alan Cooper, who, when I first phoned him, he was at Oxford, and he was moving it, and he just loved the idea, because in ancient DNA, all they'd try to get was some letters, basically of mitochondrial genome, which is much easier to get because there's many more copies of the mitochondrial genome in a cell compared to the nuclear. And they were basically trying to figure out who was related to who based on nucleotide changes. And I said, you know what I want to do, Alan? I want to resurrect mammoth hemoglobin. And he was silent for a few seconds. He goes, I love it. Let's do it. And so he was moving here. And the biggest problem with ancient DNA, the biggest problem is contamination. Because when you're working with samples that are 43,000 years old, they're broken. They may look nice. You know, you might see a cell. I have reporters phone me all the time about cloning, and we can talk about cloning. I'm not going to talk about it too much in this talk, but I can answer any questions after about that. We're not cloning mammoths anytime soon. There's so many hurdles. And even if we do, it's not really a mammoth. And they say, oh, but these cells look perfect. And I said, well, you know what? I have a 200-year-old porcelain vase on my little sideboard. I said, I don't put water in it. It's got all these tiny little cracks. That's what these samples are like. They're fragmented. They're broken. And if you get one strand with the technology we were using at the time of modern elephant DNA in there, that's what you're going to amplify. So we did all this work in a lab, and this is my student Jason. He was a summer student. He's now a lawyer working actually in the Supreme Court. He's clerking at the Supreme Court. He was a pretty awesome student, very fantastic student, and so he got to go over to Australia for a number of months and work here to ensure that there's zero chance of contamination, because that was the biggest hurdle back in 2007 when we were doing this part. So how do you go about doing this? Well, you need to know a little bit. If you're going to sequence a mammoth, you need to know a little bit about its living relatives. So the first thing is, and without the elephant genome coming out, we would never have been able to figure this out, because there's a number of problems. If you look at the alpha globin genes in our body, in mammoths' bodies, there's multiple genes, and they're very similar to one another. We've got some embryonic ones here. We've got one here, alpha D, that we don't really know what it does. Pseudogene, and here's the one that we find in adults. So we've got 6 or 7 different copies of genes that are remarkably similar to each other at the genetic level. And on the beta chain, we've got the same sort of thing. So this is the African. So we went through the genome. We found all the globin-type genes, and we annotated them, and we figured out who was who. And we determined conclusively, because we had some elephant blood, and we had mRNA, so the RNA, and we can say, yeah, these are the adult ones. And so what we did is we designed PCR primers that could only possibly stick or amplify those two genes. Because we didn't want to have any fragment from here or here, because then our results wouldn't actually be the true mammoth hemoglobins. We had to target just those genes. And again, this process seems easy for me to talk about here in 30 seconds. Yeah, that's like 2 years. Then Jason goes over to Australia and spends more than 4 months working with PCR. And I'm going to show you a little bit about each of these genes. Each of these genes, the alpha globin and the beta globin, they're not very big. They're broken into 3 little fragments. We've got exon 1, exon 2, and exon 3 on the DNA. So each of these genes has 3 little exons. And we have to target those exons because they actually encode the protein itself. So what we did is we did PCR after PCR after PCR after PCR after PCR, all in these clean labs where you have to go in and you have to disinfect and do all these standards and run all these negative controls and do it over and over again. And then we found a few changes, and then what you do is then you have to redo it and you have to clone it and you have to confirm. So here's 4 months to get 1,300 base pairs. So we had absolutely revolutionized the field at that point. You're going from 90 base pairs to 1,300. Holy smokes, 4 months only. So what did we find? 4 months of work, 7 years of background to get to the point where we can finally get these data. And I look, and we can map it here. So again, I'm going to quickly go through here. The legend, we'll go through the data here. So here we've got the Asian elephant, woolly mammoth, African elephant. So this is the woolly elephant hemoglobin gene, the alpha globin gene. And here we show with the red bars, the blue bars, and the green bars, all of the DNA changes. And if you have a thin bar, it's a DNA mutation that doesn't change the protein itself. So DNA is made into these little triplets we call codons. So there's 3 little nucleotides put together that make an amino acid. So we can have a first codon position shown here in red, second codon position in blue, and a third in green. So if we get a thin bar, we get a nucleotide change with no change in the protein. We call that a synonymous substitution. The interesting ones are these big bars, a non-synonymous substitution, because that's a DNA change that causes a amino acid change in the protein, which can change how the protein works. So I've spent 7 years, thousands and thousands of dollars. And my student was there for 4 months, and we get the data back. And we look at the alpha globin chain, and nothing. Not one single thing happened in the mammoth alpha globin chain. Fortuitously, hemoglobin is made of 2 chains. And we also have a beta type chain, which is really interesting in mammoths, and I'm not going to get into it. It's actually this weird chimera. So it's half of a delta globin and half of a beta. But we found 3 changes. All of them were first codon position, and all of them were non-synonymous. I got this data. I was sitting on my computer. It was 4:40 or 4:50 in the afternoon, and by 5 o'clock, I was sitting there by myself going, because I knew these changes were huge. I knew we hit the jackpot with some of these changes. So we've got 3 first codon position changes. And this is itself consistent with something we would consider positive selection, adaptation to a changing environment, a changing stressor, whatever it is. So what we did, the first thing we did, is because there's no mammoth hemoglobin around, there's no other things, we modeled these. So we used nice high resolution crystal structures of human hemoglobin, as it turns out, and using very computationally intensive computer programming, we could actually kind of see what these changes might do predictively. So I'm going to show you the changes that we found here in blue. The 3 major changes we found, where they're found in the protein, and I've also highlighted in red other amino acids that these ones would be predicted to interact with. And I'm going to talk a little bit about that. And this one right here, this is the one I was doing the jump about, because this one right here in the 101 position, it is a huge change where it changes a charge. We go from a negative charge to a neutral charge. And why is this one interesting? Because hemoglobin is a molecular lung. It breathes. Where does it breathe? It breathes right along that pathway. That's where it slides and moves. That's a key sliding spot. And you know what? No other mammal on Earth has that change. Well, that's a lie, but we didn't know that at the time. We've discovered one other one that I'll tell you about a little bit later today. So I knew this was key. And even more interestingly, there's a few human mutants that have that same change. So, of course, there's all kinds of variability in the human population. So some people have just natural mutations at that spot where it's a heterozygote rather than being a homozygote found on both DNA strands. And there's, well, people have studied their blood. So we actually knew these human mutants, what their effects were. And they were precisely what we would have predicted for the mammoth. And you're going, I came here for computer modeling? Did I come here? No, I didn't come here for, come on, show me the money, I hear you say, right? I want to see mammoth hemoglobin. How do you make mammoth hemoglobin? How do you make mammoth blood? Well, how you started, how we started, you don't have to do this, but there's other ways around this. But what we did is we actually got blood from an Asian elephant. And from that blood, we took out the mRNA, the coding sequence, which is just the part that actually will encode that protein. And we put it into this little circular piece of DNA called a plasmid, an expression plasmid. So we have the alpha globin gene and the delta globin gene. And the beauty of this is that this expression plasmid had been developed by a guy who turned out to become a collaborator on this project. He studied human hemoglobin. And he was interested in all kinds of human mutants and the effects of these human mutants. And it's very difficult to get blood from people that have these because there's all kinds of forms you have to fill out, and you have to find somebody from across the country that happens to have that mutation, or somebody in Israel, or wherever it is. So he developed a system where he could produce perfect human hemoglobin in bacteria. Bacteria, you think that's a little weird? It turns out a bacteria is no different than you and me when we look at them from just the molecular level. All they are protein-building factories. That's all our cells are, protein-building factories. What we can do is we can trick those bacteria into thinking this is their DNA. And they're going to say, oh, okay, I can make that recipe. And they make the recipe, and these proteins actually self-assemble. So we can make perfect, authentic human hemoglobin. So right now, all diabetics taking insulin, they have human insulin genes put in bacteria that are growing, and that's where all insulin comes from now, bacteria. It used to come from pig slaughterhouses and cow slaughterhouses. But that's not mammoth hemoglobin. Well, there's another innovation that was actually a Canadian won the Nobel Prize for, called site-directed mutagenesis. And site-directed mutagenesis is a technique where you can take a DNA strand and you can take any letter and change it into whatever letter you want. So we have an expression plasmid for elephant hemoglobin that we actually just converted into mammoth hemoglobin. So we actually have essentially mammoth hemoglobin genes put into this bacteria that we can put now into bacteria. Okay, so we've got our little expression plasmid. And I used to have a nice picture of the bacteria themselves. They actually glow red. You get these things. You have a little tube, and it's just red. It looks really quite impressive. And we make lots of perfectly authentic mammoth hemoglobin. And I know this animation isn't working. I tried it earlier. But all it shows, I can do a little interpretive dance. It just goes. It moves. Right? So we've got mammoth hemoglobin that moves when I change the amount of oxygen present. And from that, I can get curves. And what's really remarkable about this is like time travel. This blood here is no different than had I somehow got into some time capsule, went back 42,960 years, and taken a blood sample from that living mammoth itself. Absolutely no different. And when we have that, we can have those curves. So I'm going to tell you a little bit about those curves and what we found now to prove that these changes were indeed adaptive. So I'm going to show you, this figure here has an awful lot of information. So here's a typical curve. So this little dot here is the result of many, many little dots, many measurements. And I'm just going to show you something we call the P50. The P50 is the amount of oxygen it takes for half of the hemoglobin molecules to be saturated with oxygen. So 50% of the oxygen molecules have no oxygen, and 50% of them have oxygen. So it's kind of the mean oxygen affinity of that protein under that condition. So if you have a low P50, that means you have a very high oxygen affinity. So here is true data from mammoth hemoglobin, stripped. If I just take the protein outside of the blood cells and I get rid of everything else and put it in a buffer, we can measure just the intrinsic properties of the protein itself at 2 different pH values. And if I look at that versus Asian elephant, I hope you can see immediately that the protein itself, just due to 3 changes out of 287 amino acids, there's a huge effect in just its ability to bind oxygen. So mammoth hemoglobin possesses a higher intrinsic affinity for oxygen. However, red blood cells are always in the presence of about 0.1 molar chloride, right? Because that's salt in your blood. How about when we add chloride? So when we add chloride, it binds to hemoglobin and it affects its ability to offload oxygen. And this is actually really good. This helps us offload oxygen in the tissues. And interestingly, you can see here the change is much larger in the mammoth than it is in the elephant. Small change in the elephant, huge change in the mammoth in the affinity. So it has a much larger ability, stronger ability to bind chloride and be affected by chloride. There's another molecule I talked about, DPG, which is also found in the red blood cell. And when we run those curves, again, the same effect, a much larger response to this molecule found inside of red blood cells. Finally, there's protons. And since we have measured it at multiple pH values, one thing you can see here is in the presence of these effectors, the slope is higher in the mammoth than it is in the elephant. So it's able to bind more protons. It also has some other features that are different from something we call cooperativity, and that just has to do with the slope of that first curve. So I'm not going to talk about that. But again, we didn't come here to talk about cooperativity or DPG sensitivity, did we? I wanted to talk about thermal sensitivity, right? I should have a little drum roll here, but I don't have that little thing enabled. But yeah, we did find it. Yes, eureka. I knew I was right. So let's show you how I was right. And it's the first time in my career, so I like to really kind of pump it up. Every other time I was wrong on things, so it's quite fortuitous on this one. So when we just look at the protein itself, its thermal sensitivity, which I talked about this oxygenation enthalpy, just the inherent protein itself is no different than that of elephants in its temperature sensitivity. But if I add chloride, we have a much stronger effect in the blue bars, the mammoth, than we do in the elephant. And when I add that other molecule, which is always found in the red blood cells, BPG, it also has a larger effect. So both of these independently contribute, and they're both significantly different than the effect of that same molecule on Asian elephant, African elephant blood. So the reduced thermal sensitivity of mammoth hemoglobin arises from 2 separate factors, chloride and DPG. And we can even look at this. We've done many more curves that are still unpublished right now in these things. But we can see here, again, this is the log chloride concentration. So as you have more and more chloride versus the log P50, all we need to look at is the blue and red. So we can see blue is a much higher slope, very small slope for red. And we can even calculate here how much chloride. So we can see mammoth hemoglobin, each molecule binds 1 more chloride molecule than does the elephant. And we can do the same kinds of curve for DPG. And as it turns out, mammoth hemoglobin binds DPG a little stronger. So when we're looking at the population, we have more DPGs bound at any given time to the mammoth than the elephant. Oh. And what I find truly remarkable here, when we look at our final value here, around 17 or 18, it's almost identical to that of reindeer and muskox, the two other truly cold adaptive hemoglobins that are found. So they've come down convergently to the same solution, to the same level, the same thermal sensitivity. It's a wonderful example of evolution. Again, convergent evolution, because we know mammoths are not at all related to reindeer and muskox. And we can even take this further now. Why just look at physiology when now what we can do, we've got mammoth hemoglobin, and we've got a change not found in any living species. Well, let's look at those changes at the atomic level. I'm talking at the angstrom level. Whoops. I didn't want that guy to come up. So I'm going to talk about, just here, I'm just going to focus on that one change found in the sliding interface, just to show you how we can now take this a step further, this kind of work. So again, we have a negative charge being turned into a neutral charge right in the middle of a protein that moves right on that axis. And I'm going to show you at 1.5 angstrom resolution. Here, if we look at green, this is the mammoth. Here is human. And this is not residue 101, it's residue 104. Because residue 101 here interacts with 104. 101 and 104 interact in blue. Right? So we see the human here. Well, as it turns out, in the mammoth, this 104, which is arginine, a very strong positive charge, is free to move elsewhere. So we get a radical change in the position of this arginine at 104. And what this does, and we can even actually explain our results. Why does mammoth hemoglobin have such a higher affinity than Asian elephant hemoglobin? Well, it's kind of like we've introduced a magnet or a pair of magnets. By getting rid of that positive charge and allowing that negative charge to move right into the middle, we've got two strong negative charges sitting right beside each other, and physically they repel each other. So we're destabilizing the the low affinity state. So it explains our high affinity, and at the same time, it has effects on two other molecules, which are lysines, which are also very strong positive charges. And it makes a nice binding site for a single chloride molecule, precisely in alignment with our physiological data. So this is actual crystal structures at angstrom resolution of mammoth hemoglobin. So here's the last change, the last slide on this, the take-home message of this. What is the effect of this? What is the benefit of this hemoglobin? What does it really change? So if I looked at Asian elephant hemoglobin, and this is stripped all by itself, we see the mammoth change causes the protein to have a much higher oxygen affinity, right? Because we destabilize that tense state, that low affinity state. And this change itself would be hugely maladaptive, because we've got hemoglobin that binds oxygen so tight that it can't release it. But this identical change causes another effect. It causes more chloride binding, more DPG binding, which in fact stabilize the opposite state, the high affinity state, the low affinity state. So we see here when we add chloride and DPG, the curve of the elephant gets shifted over to the right, and that of the mammoth is perfectly overlain over it. So what this tells us is at 37 degrees Celsius, mammoth blood was absolutely no different than elephant blood. But at cold temperatures, mammoth blood responded less to temperature, and it was able to offload more. So, in the rest of the body, it has no effect. It only has an effect at cold tissues. Such would be experienced in the winter. Right? And it does this, so it changes the ability of hemoglobin to offload in the cold, but has no effect in the warm. And here's even a crazier thing we were able to figure out. Every single conceivable other change of that position would have been maladaptive. This is the only possible solution. Any other amino acid change there, you would get this shift to the left and no corresponding shift to the right. So they picked the only possible solution. And to me, that's pretty impressive. And again, it was even more impressive at the time that no other mammal had ever shown this kind of change. But that was about to change. And again, this is test tubes. I'm not really working with a mammoth, right? As awesome as it is for me to do this kind of stuff as a physiology, it's still a test tube of bacterial hemoglobin. And we thought, there's no way. As I told you, you've got a block of ice that's been sitting there for 40,000 years. I had some hamburger from my freezer from last summer that we pulled out, and it was brown, and it doesn't smell right, right? Imagine 40,000 years of freezer burn. Well, last May, I received an email from this gentleman here, Semyon Grigorov, and I was in the middle of something, and I got this email, Mammoth, blah, 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 read your paper, blah, blah, you know. And I went, oh, okay. And I went back to my job, doing things. And that evening, I came back, and I was going, Mammoth discovery, you know, and he was telling me about this mammoth. And so this is, you know, one of the early images, video. This is basically what they found. This, you know, you're looking and you go, that's a mammoth? Okay. It's this tiny little island, fairly close, but even further north than the specimens we used. And I got home, and a mammoth, brand new discovery, and he's talking to me about how great this discovery is. I was going, okay, so great. So I go on the internet, nothing. Huh? And as it turns out, you know, why did this guy email me? Well, because in this specimen, he also sent me this picture. So I got home and I looked at these photos, and I went, what? Holy smokes! And he's talking about the blood, and they found liquid blood. Look at that. Yeah, that's liquid blood. I work with blood. That's liquid blood. I'm sure it has other things in it, but that doesn't really matter. That's liquid blood. And then I was going through his email again, reading it carefully, and like I said, I find nothing interesting. I was going, nobody knows about this. This thing was just discovered. This guy just got back. And they found blood, and I guess he Googled me and found our mammoth work. And do we want to collaborate? Of course we want to collaborate. Are you kidding me? So I was invited in August to go on a follow-up expedition. And what you saw before, right here, I mean, that's not too impressive. But as it turns out, this mammoth was encased in, not permafrost or anything, but ice. And this ice itself makes it a barrier for oxygen and all kinds of other nasty molecules to get in. And the preservation of the stuff underneath is unbelievable. Take a look at that nose. I mean, we don't have mammoth trunks like that. That's incredible. Look at this muscle. That is pink. That's not freezer burned. That is fresh meat. This animal now, we just got the results back, 43,000 years old as well. And here, they cut up a bone. That's some bone marrow. That's still yellow bone marrow. This is just unbelievable. So I was invited to this, and as it turns out, I had a collaborator coming from Japan. We had holidays and everything. I said, oh, I can't go. As much as I wanted to go way up there and see this thing, I just couldn't make it. And he said, don't worry, we're going to have a follow-up in spring. Whoops. As it turns out, that was like 2 weeks ago. And they invited me the day after I booked a whole family vacation and another collaborative thing down to California. And then when they invited me for the start of March, I quickly looked at the weather, having spent winter in Winnipeg. And Palm Springs, plus 28. Yakutsk, minus 36. And I went, yeah, I'm not changing the travel plans. And when I asked, you know, just a couple of weeks ago, I asked Francois, I said, is there any chance I can change my talk to include some of this stuff? Because I was going to talk about whales and myoglobin, and it's another fantastic story. And he said, oh, this would be so great. He's always wanted to see a mammoth autopsy. And I got the very first images, and this is from March 2013. This is the last thing I thought I would ever see. And here's Semyon right here, and I'm going, is this like what you guys are doing? I'd like to see some of the backroom stuff. Look at this crazy big drill. They've got the thing here. And since I couldn't make it, and as Francois mentioned, I did a postdoc in Denmark. And who did I work with? Well, it turns out I worked with Roy Weber. So I called Roy up and said, Roy, you know, look at this. We have an opportunity to get actual mammoth blood and muscle to look at myoglobin and some of the other things. And Roy has worked on hemoglobin for over 50 years. He's like the world's authority, I think, on this. So he's going, once in a lifetime opportunity, and I couldn't agree more. And so luckily enough, Roy got to go and sample. And here's Roy taking actual, it's hard to see in this image here, but that's a big blood vessel. Can you guys see that blood vessel right there? You see that little hole? It comes right through there. So he was actually to take out, and get this, there's intact red blood cells. Intact 43,000-year-old red blood cells still in there. The muscle was perfectly pink, and we're moving this. And so you see, we're shipping these back to Denmark, and we'll be working on them this summer to get, to really verify and confirm our results. Because it's one thing, again, to get something from bacteria. And who would have ever have dreamed that we can get physiological measurements from something that truly died 43,000 years ago? That's pretty impressive. So that's one part. And I'm going to talk now a little bit more about some other advances in the field. So again, we now have the opportunity to look at these things from a 40,000-year-old animal. But changes in ancient DNA technology, in just biotechnology, what we can do are outstanding. And we're interested, as I mentioned, in this other group. So after we got that 4 months to get a few DNA samples, we want to work on these guys, stellar sea cow. Again, they evolved from a tropical environment to an Arctic environment fairly recently, and they were huge and weird. And this is just a gorgeous picture by Carl Buell. I think it's up in the Smithsonian. It was just put there a year or so ago. Where were these guys found? Well, they're found way up in the Arctic here. And so this is the Commander Islands, but they came across all the way to Alaska as well in the archipelago. And what's interesting about these animals compared to their relatives, we see in the news every year about manatees dying of cold. When water temperatures drop below 20 degrees Celsius, manatees die from cold. This is an animal that water temperature, because it's salt water, would go down to minus 1.9 degrees Celsius and probably never go above 5 or 7 degrees Celsius. So these are great animals to look at cold adaptation. They also have no fingers. They're the only animal to have completely lost their fingers. They lost their teeth. So there's all kinds of really weird things about these animals that we want to look at. But I don't want to spend 4 months on PCR and contamination and all these issues. And now, as it turns out, with this new technology, this work can be done in your labs here. And I'll tell you a little bit about this new technology that we started using, and it's just moving like crazy right now. So here's some stellar sea cow bone. And what happens when you extract DNA from a sample that was collected in the 1800s? Well, you know what you get? You get a lot of bacteria and fungi. That's basically what you get for DNA. You know, between 90, 99, or 99.9% of all DNA from a mammoth bone is bacteria and fungal DNA. And in there, you get a little bit of, stellar sea cow or mammoth DNA, just a little bit. But I'm not interested in 3.2 billion base pairs. I'm interested in, 800 base pairs, some targets. Even if you look at 100 genes, we're looking at 0.001%. If I look at the entire protein coding region of the animal's genome, that's 1% of this 1%. It's a very difficult thing to try to find these things and get these. And that's why, turns out, very few ancient species they've ever tried to do genomes from, because it's so expensive, and basically you just get genomes of bacteria. Hugely expensive, all kinds of problems. That was then. Now, there's a new technique. So what they do is we can take all these little tiny fragments and we put them into what's called adapter sequences. And we make a library. So we get all these ancient DNA sequences, and they simply get a DNA strand on either side added to them. And what's nice about this is it's an archival permanent sample, because we can we have these green regions here, you can make primers, and if your sample gets lower, well, you just make more. You don't need ancient DNA. And once it's put into these guys and you make this little library, I can send it to any lab in the world, and you don't have to worry about contamination because of these little blue bars here. I'll talk about these little blue bars in a minute. So you don't have to worry about contamination anymore, and you don't have to worry about running out of sample. What's even more amazing is what we can do with this now. Instead of doing PCR to get 90 base pairs or 100 base pair little fragments, what we do is we throw all of that DNA into a little test tube, all of it. And then we add what are something called biotinylated little oligos. And all these are tiny little DNA strands with a bit of biotin here in yellow. And these are designed to be complementary to our targets. So basically, if I'm looking at the alpha globin gene, I just take the complementary sequence, and these guys would naturally just match. And what we can do then is add some magnetic beads to the solution and pull out only the blue targets that we're looking for, and all the crap, everything we're not interested in, simply gets thrown away. We take every one of those individual DNA strands that we're looking for, and we simply just release them from that magnetic bead, and we put them into these brand new mega sequencing machines. They're just unbelievable, the data they can put out. Complete genomes now at high coverage in 10 hours for humans, for like $1,000. So we started this, and here's my PhD student, Anthony. He's in an ancient DNA lab. He's going into the extraction room. So we did this. We made these special libraries for a manatee, 2 dugongs, and 3 stellar sea cows that we got from a museum in St. Petersburg that were collected in the 1880s. And again, we did this and to further get more data at less expense, because expense is the biggest hurdle for most labs. Again, this is where these blue bars come in here. What this is a 7 base pair sequence that we call a barcode, an indexed barcode. So for each specimen, when we make the library, it gets its own unique barcode. So, Stella number 1 gets barcode AAG, TTT, and the other one CCC or whatever. So you can make all these kinds of libraries. And the beautiful thing with this is the sequencer reads that barcode. So we can throw 3 animals in together at the same time. You can never do this with PCR because you get in all kinds of mixing up and all kinds of stuff like that. We can figure everything out that way. And so we targeted it, instead of 1,300 base pairs over 4 months, we targeted about 200,000 base pairs, 100 genes that we're interested in, which equates to about 0.006%. 4 billion base pairs of this manatee genome. We ran them in on a machine. This is a couple of years ago already. And we're still working through the data. 2 lanes on this Illumina GA2X, which is an older version now. And we got back 87 million sequence reads. And they're each about 54 base pairs long. The total amount of data we got back was more than a complete genome. 4.7 billion base pairs is what we got back. And this took 3 days. 4 months for 1,300 base pairs. 3 days, we get from 6 animals a complete genome, more than that. That was a couple of years ago. The new HiSeq X, right? We got back 87 million. It now puts out 3 billion reads, 3 times longer, 1,700. I mean, you can do dozens of genomes in a sequence. It's just crazy, this technology, what kind of data you can get with it. And instead of doing these tiny little fragments, what do? Well, we get it all back, and it's all computer animated now. We can simply put some formulas in, and we say, I want to take the manatee the beta globin gene, which we see the coding region, exon 1, exon 2, exon 3. This is the data we got back from the stellar sea cow. And as it turns out, it went down here somewhere. I cut it off just so we could see. That's every single fragment of DNA we got back. And then we can look at it closer, and all these alignments. And sure enough, when you see here, again, the top here is the manatee. This is a stellar sea cow. So we can see there was an A to G change here, a C to G change here, A to T. Right? So we can see those changes. So stellar sea cow in exon 1 has 4 changes relative to that of the manatee. And another beautiful thing of this system is because we're sequencing every single DNA strand individually, we don't have to worry about all these PCR artifacts. There's no worry. Even if we get a little bit of contamination in there, that's going to be so swamped by all these reads. And we can even pick up all the ancient DNA damage. So ancient DNA that's been sitting there for a while gets broken down. And C's sometimes get turned into what looks like a T. G's get turned into A's. But you know what? We can identify all of those, and we just throw them out. So we can get perfect sequences from all these individuals. And just to show you the quality and the amount of data that we can get with this approach, 26 of the genes we put on there were something as part of the Assembling the Tree of Life project for mammals, which is looking to get the phylogenetic relationships of every mammal. So these are the genes they're using. We put on 26,000 base pairs of genes here for that. And when we look at this, and we made our probes or oligos based on living species that were closely related. So most of these we were able to get dugong, which is the closest relative. We targeted 17,000 base pairs. We got 17,000. 99% of our target at deep coverage. When we used manatee, 98%. Even when we used an elephant, an African elephant DNA as our template to try to catch something, these guys diverged 65 million years ago. And we're still able to get 99% of our target from something that diverged 65 million years ago. That's just totally impressive. So when we're doing this for hundreds of genes, as it turns out, all kinds of proteins, all kinds of things, they don't work in isolation. They all work together as teams, as networks, developmental networks, all kinds of gene signaling pathways. But we don't have to look at one gene. In fact, we can get the entire gene network at one time. So I just showed you a little bit of data on the beta globin gene. So before we started this is what we knew about Cyrenian alpha globins. We knew not a single sequence. And just through some of the previous work we did ourselves, we had 2 genes from 2 species. This is what we do. 3 days later, we've got everything, complete, all of them. We've got the embryonic genes, we've got the fetal genes, we've got the adult genes. And as it turns out, we can express them all in bacteria. And we can look at maternal, fetal, oxygen exchange, all kinds of things in these guys. And we found some really interesting things already with the Cyrenians. So I showed you this data quickly. When we looked at the mammoths and the beta globin, we found 3 changes, 2 in the African elephant, 5 changes in the evolution of these guys. We'll take a look here at these guys. Well, what is that? 37 changes. Huge number. So we know these guys have undergone massively rapid evolution of this gene. And we can see the amino acids from all these genes. And we know for a fact now that the stellar sea cow also has magnificently awesome hemoglobin, not found in any living species. Why? Because of this change here at position 82. That is right in that central pocket that binds DPG. So these guys could be totally radically different. Right? So that change here is found right in this pocket where this DPG is. It makes another charge change. So we know the stellar sea cow is going to have crazy, weird hemoglobin. And get this. Remember I told you I lied about that mammoth change? Well, there is a living species that has the same one, the dugong. The dugong has that identical change as found in the mammoth. But they're also even more wacky. Because I mentioned this 101 interacts with position 104. Well, that's also been changed in the dugong. So we actually don't even know what this change is going to do. We can't even predict that. And there's another, this guy also interacts right here with 94. So we're expressing all these guys, and we can take it even a step further. This is a whole new area, too, that we can expand into. Since we have all the tips of all these branches, we can do something called ancestral sequence reconstruction. We can go back in time. I said we all have an archive of our history. So when I look at all the similarities and differences between species, we can go back in time and we can determine the precise protein sequence of the ancestor of dugongs and stellar sea cows from 23 million years ago, and we can express their hemoglobin, or their insulin, or their uncoupling protein 1, or their eye genes, or their hearing genes. Or we can even take it another step back because, right, and we can resurrect these things, and we can measure them from an animal that died 23 million years ago. We can get those living type measurements. And we can take it another step back, and we can go to the ancestor of all of these guys 29 million years ago. And right now, there's work going on to get basically complete genomes of 200 extinct species and 97% of all living species. So we can go back, as it turns out, 200 million years ago. That's what we're going to be able to do in the future. We're going to be able to express hemoglobins, eye genes, all kinds of things from animals with precise certainty, confidence that that was the genome from an animal 200 million years ago. And we can resurrect those traits. 200 million years. I mean, that's completely amazing. Dinosaurs, unfortunately, we can't do that. We can just go back to the ancestor of birds. And very soon you're going to see the 30 bird genomes coming out. So but we're going to have to fill that in, but you can do the same sort of thing with that. So technology now, I mean, the amount of data. All we're doing here, again, is we're just looking at the tips. But every single branch we go back, we've got that data. So again, it's mountains. It'd take 1,000 careers to just start to look at some of these genes and some of these interactions. But that's what the future lies for, the interface between genetics, physiology, all these things. Oh, I'm probably over. Yeah, I was going to say, okay, sorry about that. I won't even go through this, but we can even take the steps further, not look between species. We can look within a species. I'm going to go through this super, super fast. When we look at mammoths, it's a hugely diverse lineage. We've got hundreds and hundreds of mammoth specimens that are yielding DNA. Right? We don't even know the relationships. The steppe mammoths, are they here? It looks like Colombians might be here. There's all kinds of phylogeography within this lineage. We've got different ice age. Here's the temperatures over time. We can go back. We can get data from all of these animals. And again, we can do the exact same thing within, let's say, just the mammoth lineage. We can look at population genetics of animals that, from individuals 50,000, 100,000 years old, 100,000 years ago. Again, we put our strands in. We have all these indexes. With this technology and these sequencing, we can run 200 mammoths at the same time across 100,000 years or more of evolution. And we get any kind of genes we want from them. And we can make these trees, and we can map everything. So not only are we looking between species and going within certain lineages, we can look at 100,000 years of evolution in real time. Looking at all these specimens. And this is, this tree already here is a couple of years old. So there's hundreds, several hundred mammoths right now that are giving up their DNA that we can look at this and find specific changes in certain lineages and correlate these with temperature, where they were, climate, you name it. So that's the future for this field. So with that, I thank you for your devout attention, and I tend to get really excited and talk about things, so sorry for going over. And I'd like to thank, of course, again, this work couldn't have been done without some of the students, collaborators from all over the world. This was the work on just the mammoth hemoglobin itself, and this is some of the more recent stuff with the stellar sea cows and such. So I thank you again for your attention. You should let me know. I didn't even look at my watch.